Okay, excellent. So this talk is kind of slightly unusual in my sort of range of talks I do because it's been I've been using invertebrates almost for around 18 years, but now I'm a lot more into invertebrates. I recently set up a special live invertebrates laboratory. So before I will. I'm sorry, I'm struggling with switching slides now. Oh yeah, okay. So before I will move to invertebrates, I just want to briefly explain that how we test our polymers or any other materials. We clearly use various techniques. Some of these techniques are in vitro techniques when we use cells. And this will be cell culture. This is the most common nowadays, to use various cell culture techniques to characterize toxicity. Sometimes we use ex vivo models, and ex vivo models are very popular in my group in particular. So we work with the animal tissues. Typically, we receive them from the local abattoir, like cow's eyes, for example. We bring them to the lab, and then we study various interactions with polymeric dosage forms, such as adhesion and penetration. In more rare cases, especially nowadays, we use in vivo models with mammals. And obviously, there's lots of ethical dilemmas by using live animals like mammals, rats, mice, and rabbits. Therefore, scientists who work in this area, they started thinking about animal experimentation. And just to give you a bit of statistics, that, for example, only in the UK in 2023, there were 2.68 million procedures conducted on live animals, and these are mammals. And out of this, over 80,000 of these procedures were assessed as severe and causing long-term disease, and obviously clearly cause lots of suffering to animals. One of the really well-known tests in this area is so-called Drey's test. With the rabbits picture, you could see when Drey's proposed a test either to use rabbits for testing ocular irritancy, to instill some formulations that could be nasty to rabbits' eyes, and observing the changes happening. Often these tests causes blindness and pain, and this test has been heavily criticized. There were lots of replacement strategies. Another test that is quite cruel is typically used in wound care characterization. So it is then in rodents like rats, when an artificial wound is created, and then we could put some of our formulations, for example, hydrogel wound dressings, on the top to test antimicrobials. And then we monitor how the wound will be closing. So clearly these tests are very cruel, and I've been thinking and have been analyzing literature what could be done in place of these tests. And scientists working in this area, they proposed so-called 3Rs approach, and 3Rs stands for reduce, refine, and replace. So if we could reduce the use of reduce the number of animals used, then we could greatly benefit these animals. We could also refine the procedures. For example, when we use, let's say, local anesthetic, we could refine the procedure, we could reduce suffering, or we could use replacement technologies when we could replace animal models either with some computers, some computer modeling, or with lower animals, or with synthetic models. So in this talk, I will be talking about replacement technology when we are trying to replace the use of mammals in animal experimentation using live invertebrates. But then before that, I would like to talk about what are the protected animals, especially in the UK legislation. So all living vertebrates, such as fish, amphibians, reptiles, birds, and mammals, they're considered protected. So in order to conduct any laboratory experiments on them, you need to apply for several types of licenses. Additionally, cephalopods, which are invertebrates, but because there was an evidence that cephalopods, they have like a more developed nervous system, so theoretically they could sense pain, therefore they are also protected. And specific example also in fish, experiments with fish, especially zebrafish, it's a very common model. Zebrafish is obviously a vertebrate, so it is protected species. But then if you would like to conduct experiments in zebrafish larvae, especially just a few days after they're hatched, until they will start independent feeding, they will be classed as nearly invertebrates. So you could do experiments with them without applying for any licenses. But then non-protected species are invertebrates, and the most common examples will include insects, arachnids, gastropods, crustaceans, and worms. There's lots of examples in the literature. For example, on the picture is Drosophila. Drosophila resulted in several Nobel Prizes. And another example is the worm, is C. elegans on this slide, also several Nobel Prizes. When I came to study the life and to study drug delivery systems and to develop drug delivery systems, I started using slugs. But now in my lab, or the lab I established recently, we use slugs, we use planaria worms, we use the bugs moth larva, and also Daphne, and I'm looking forward to expand the portfolio. So I will tell you a little bit about some of these models. So initially, when I started my independent research career at Reading University, I came across this study from University of Ghent in Belgium. And this study, they were reporting the use of slugs as a tool to screen irritancy potency of different chemicals. So I came across this study and I got really interested, and I started developing this capacity in my research group. And over these years, we've sort of refined these tests, and this test is very simple. You need to have slugs, you put them on a surface. If the surface is irritant, then slugs will release mucus, and you could measure this mucus gravimetrically, weighing slugs before contact, keep them in contact with your formulation for 1 hour, weigh slugs, wash the mucus released, weigh them out again, and quantify the amount of mucus production. So obviously, as in all biological experiments, you need to use controls, positive control and negative control. So typically what we do, we use benzalkonium chloride. 1% solution is a severe irritant, so mucus production is very high, is around 30%. If you use phosphate buffer, then the mucus production will be around 5%. So if your formulations or polymers are between these numbers, then then they could be either irritant or non-irritant. And we have done lots of studies. Well, some of these examples, some of our studies are listed here as publications with slugs. And to characterize slug mucosal irritation tests, this could be used for testing irritancy, and both solutions, films, powders, and semi-solid formulations could be used. I'd like to point out that some of these formulations cannot be tested in cells. For example, you cannot test films until they are hydrated in cells, but you could test them in slugs. The test is very simple. You could generate data quickly. Slugs are very sensitive to irritation, and you don't need specialized equipment, just analytical balance and some containers for keeping slugs. But then there's always limitations, such as difficulties to breed slugs. Well, we've tried, like around 18 years ago, we tried to breed slugs in the lab. Also, I was trying to breed them at home, and it takes ages and resources, and you need to take care of them, cleaning and everything. Therefore, with all these years of experience, we switched to catching slugs, to collecting slugs in wild. And this is the biggest limitation of this test, because the seasonal availability of slugs in the UK, typically it is from May to September, and also it depends how dry the weather is. So if, for example, this year, the weather in the UK is very dry and we're lacking slugs. Well, we've been using slugs for many years, but then because of these limitations, I got interested in another model. Another model is worms, called planaria, and they are aquatic worms. They're non-parasitic, they're flat, and they have lots of interesting characteristics, such as they have excellent ability to regenerate their body. If you cut them into different pieces, each piece will regrow into a worm, into a whole worm. They also have a pair of eye spots. They react to light, and they are established animal model in biology and pharmacology. There's lots of studies, both in regeneration medicine and in psychopharmacology. And we decided to develop irritancy test based on planaria. So we tried different approaches, such as looking simply at the viability assay when we are putting planaria into solutions of different chemicals. We identified chemicals with known irritancy status, and basically we were studying them systematically at different concentrations and different exposure periods, monitoring how quickly it will result in planaria death. What we found that was some correlation between the irritancy potential of these chemicals and viability, but we were not really satisfied with this correlation. Correlation was not so good. So we tried another test. Another test is based on the ability of planaria to move. And you could see this video. If you put planaria worm in a petri dish over the graph paper, and if you count the number of crossings in a graph paper, then you could quantify the speed quickly. Their speed of movement, well, you get a value, not exactly the speed, but a value that will be proportional to the speed of a worm. And what we found that the worms, they will respond to different chemicals. Again, the same range of chemicals we used for testing. And we've established that there was some reaction of worms to strongly irritant chemicals, but not so much to weakly irritant chemicals. So we then, after this experience, we developed our own fluorescence test. And this test involves exposure of planaria worm to a solution of the chemical of interest. If the chemical of interest will be causing irritation to planaria, then it will be breaking their epithelium. So what we need to do, we need to expose it to the chemical of interest, then move planaria into artificial pond water to wash this chemical off. Then we place this worm into a solution of fluorescein, and if the epithelium is damaged, then fluorescein will go inside the worm. Then we wash the excess of fluorescein off, and we immobilize the worm using agarose gel or gelatin, putting it into ice and taking a fluorescence image. You could process fluorescence images using the ImageJ software, and you could generate fluorescence intensity values, and we could see that at least this test could predict the strong irritants. We have a strong response to strong irritants. And we've used this test for a number of polymer-based projects. Well, just to give you one example here, in a publication when we did chemical modification of hydroxyethyl cellulose by introducing maleimide functional groups to increase mucoadhesive properties, and we used fluorescence tests in planaria to establish that the HEC with limited number of maleimide groups doesn't cause any irritant properties. So to summarize about the planaria, and planaria tests could be used to evaluate skin and mucosal irritancy, could be applicable for liquids and semi-solid formulations. You can't use them for solid formulations or films. Planaria are very easy to breed in the lab, and in our newly established lab, we have lots of fish tanks with planaria. They eat meat, they eat liver. Different types of assays could be developed. Well, I demonstrated viability behavior. We also are looking at regeneration potential, and epithelial damage assay is based on our fluorescence. And there's also possibilities to study chronic exposure, which you could not always do with cell cultures. This model, as a limitation, this model cannot be used in solid state, and further validation will be required, but we are working on that. Our next model, we started it just last year, is called Galeria melanella. It's wax moth larvae, and we've recently established the full breeding cycle, but Galeria melanella in a larvae stage, they are available commercially in the UK at least. They are used in pets industry to feed different pets, such as snakes, lizards, and frogs. But we established the breeding capacity in our lab, and we've established that the Galeria melanella larvae bred in our lab, they show slightly different properties compared to commercially available ones, because what we know that commercially available, these commercial companies, they possibly use hormones and antibiotics to breed them. And these are the results of the studies of a few of my project students this year. We tried to generate LD50 values in Galeria melanella. And for those of you who are not familiar with LD50 is lethal dose, that minimum concentration of a chemical that will cause 50% of death in an animal. So we've correlated LD50 in Galeria, and we took literature values for LD50 in rats. And as you can see, there's some relatively good correlation. Of course, to establish it further, we need to characterize a few more compounds, but I'm sure that we will do that in the nearest future to establish a good correlation. Another really interesting test where Galeria could be used is infection tests. So Galeria, they have innate immunity that has some similarity to human and to mammals. So what you could do, you could inject bacteria into Galeria, and if you inject bacteria into Galeria, they could develop infection, resulting in a larva death. And depending on the concentration of bacteria you inject, the death could happen really quickly, or you could postpone it. But then after infecting Galeria, you could also treat it with antimicrobials by injecting antimicrobials, and in some cases, you could achieve a full recovery of an animal. So that's what we do. We inject Galeria with Staphylococcus aureus, and then if you don't treat them, then they die and they turn into like dark to black colors, and this process is called melanization. And so when we were looking at this model, we picked it up from the literature. We started looking at optimization. So we were injecting different concentrations of Staphylococcus aureus. You need to identify the suitable concentration to have the infection controlled. For example, if we inject very highly concentrated bacterial suspension, then all larvae will die in 4 hours. If you start reducing this concentration, then the larvae will survive for maybe for a few days. And also, this is an example of treatment larvae. Well, in these experiments, we use chloramphenicol, and chloramphenicol is antibiotic. And depending on the concentration of chloramphenicol, you could observe a partial recovery of infected larvae, but also you could observe even a much worse situation. You could increase the death rate of larvae because of some toxic effects of chloramphenicol onto the immune-compromised animals. And this is also mirroring the situation with chloramphenicol even in human, because chloramphenicol is only used in ocular drug delivery as a topical antibiotic, and it's never been, and now is not used in like injections or orally because of some toxicity issues with chloramphenicol. So we have started doing disinfection models with a few polymer-based projects. Well, in my group, we attempted to synthesize the antimicrobial polymers. For making these polymers, we were making them from commercially available poly-2-ethyl to oxazoline by hydrolyzing polyoxazoline to generate linear polyethylenamine, and then by reacting linear polyethylenamine with bromoethane to produce quaternary ammonium moieties. Obviously, we've done all these sets of characterization to prove that this quaternary reaction does work. And we started treating our infected larvae with these polymers. Well, we did use several polymers, and we also work with quite a few collaborators who provide us some antimicrobial polymers. And so what we observed that in some cases, we do observe the improved potential for larvae survival when we inject antimicrobial polymers. But in other cases, well, sometimes polymers could be also toxic to the infected larvae, even though the polymers, when we inject them into the healthy larvae, larvae may survive them. Another interesting model which we recently started developing is the wound burn models. Sounds very cruel, but we use like some really hot metal rod to cause a burn in a healthy larvae. And once the burn is created, then we did lots of control experiments where we created the burn without infection. Also, we induced the burn, and then we introduced infection. We introduced Staphylococcus aureus, and then we tried to treat these infections with some polymer-based formulations. For example, we treated them with iodophores. Currently, in my group, we are studying iodophores, and one of the iodophores is a complex of polyoxazolines with iodine. So what we've established that one of our iodophores based on polymethyloxazoline mixed with iodine, it actually could improve the survival of the larvae after infected burn is created. Is relatively early results generated by master students this year, but I'm really looking forward once we will generate a few more data, then this will be a good validation. I found this model again in the literature. It's not my invention, but the model has been reported a couple of times in the literature, and we are basically trying to develop it further in our research group. So the advantages of Galerian, we have potential for evaluating toxicological properties of chemicals in healthy and infected state in larvae. You could also use this larvae in the studies of antimicrobial properties. There's lots of interesting features of these larvae. Specifically, these are one of the few who could survive human relevant physiological temperature. You could keep them at 37 degrees. You also could use 3 routes of administration of compounds. You could inject your chemicals, you could force feed your chemicals into the larvae's gut using the same needles, microsyringes, and you could also apply your chemicals topically. There's possibilities for calculating LD50 values, and we did observe some very good correlation, and sometimes the LD50 values in larvae were matching those reported in the literature in rats. Of course, these toxicological models do require further validation, and we're working on that. One of the limitations, especially for commercially acquired larvae, they are prone to fungal infections. So you need, when you get the larvae, you need to basically to do a bit of selection and identify larvae that were infected already, so we discard them. And also what we know that commercially available larvae, they're possibly treated with hormones and antibiotics. So we are developing and developed our own larvae. And it's relatively easy to breed them. What we've also established that there's a difference between commercial larvae and in-house bred larvae. So when we will be reporting it in our publications, we will be specifying which larvae we have used in our experiments. Well, to conclude, I hope I convinced you that live invertebrate tests do have some potential for toxicology and biological activity testing, and huge potential for testing various polymer formulations. We recently established PVTAC as a physicochemical ex vivo and invertebrate test and analysis center at the University of Reading. You could scan the QR code here and you could follow us on LinkedIn. We are not the company yet. center, and we at the university, and we do work with various industrial partners. We develop new toxicological and biological testing screening tests, and we're happy to collaborate both with industry and academia. And final slide, I need to acknowledge lots of people, especially those whose names are highlighted in red. And those people contributed to various parts of these studies. And on the picture is a part of my research group. We recently did exhibition of our invertebrates at the University of Reading Community Festival, which did receive lots of interest from general public. Thank you very much.